Thank you, Dr. Sambo. So I am really thankful to the organizer, particularly Dr. Sambo, for inviting me to be here for this important conference. So at this symposium, the clinical pharmacology and patient symposium, clinical research and ethics. So we have three eminent speakers at this symposium. Number one speaker, Pharmacogenomics to support drug development research, and it will be delivered by Professor C. Adithan. The organizer feels honored to honor Professor C. Adithan to be with us online mode to deliver this important topic, pharmacogenomics to support drug development research. So we need not to introduce Dr. C. Adithan. He is the eminent scientist in the field of pharmacology, not in India, in abroad also. So I will request Professor C. Adithan to deliver the lecture.
Sir, will you share the screen, sir?
Okay.
Or otherwise we can share, you can tell us to change, we can change that. Hopefully you can see this, our screen, sir.
Okay. Kindly you share the screen. I have some technical difficulties.
We are sharing, sir. You will tell us to change, we will change, sir.
Okay.
Thank you, sir.
So I would like to thank the organizers for giving the opportunity. Dr. N.R. Biswas, then Dr. Tripathi, and I am also able to see many of my old colleagues. Now, next 15 minutes, actually this has to be a 1 hour topic, but I will try to do justice within 15 minutes. This is the pharmacogenomics to support drug development research. Next slide. Next slide. Actually, in this 15 minutes, I am going to cover the time factors, cost factors in drug development overview, then the incorporation of pharmacogenomics in various phases of drug development. This is what I am going to attempt these 15 minutes. Next one. Next please. Yes. As you all know, BMS drug development is a laborious and time-consuming one. We start with 10,000 compounds and we left with only one approved drug. Next slide. And it takes about 12 years to complete to bring the average NCE to commercialization. Then nearly 1 billion. Nearly 1 billion we have to spend. And it happens, $200 billion attributed to compound failure. Next slide, please. The cost of fundamental and clinical testing for MC is, you can see here as it is shown. The phase 3 is highest. It is 27.1. The lowest is animal model testing. It is 1.66 million US dollars. In case, let us say, next slide. Next slide. In case there is a termination of drug clinical trial in the phase 2 or phase 3, it costs lot of money. Therapeutic diseases due to lack of efficacy and safety concern. So we have to take care in designing the clinical trials, recruiting the subjects so that the study cannot be aborted at the later phase, either phase 2 or phase 3, which will be very, very expensive for us. Next one. Next one. This slide you can skip. Next one. Next. So first one is drug target identification. This can be done by in vitro study as well as the animal study. Here, if we can say, all of it are aware that drug target may be enzyme, or a receptor, or transporter, or a protein in signal transcription, or any protein produced in catholic condition. It happens that proteins, that is a receptor, transporter, are polymorphic. They are controlled by gene. If there is a polymorphism in the genes encoding these proteins, then there will be variation with that. For example, polymorphism of beta-2 adrenergic gene and the P2Y12 for receptors can lead to inconsistent result in preclinical and clinical studies. So we have to be very careful. Thus, at an early stage, the target can be characterized based on pharmacogenetic studies by in vitro or by animal studies, compared with the proteomic and suitable drug compound in selecting the further studies. Next slide. In phase 1 study, with all of it know that the main purpose is to establish the safety of the drug. Here we include the subjects, healthy volunteers, but to say whether they are healthy or not, we have to do pharmacogenetics testing, ideally, because majority of the problems are come due to the pharmacogenetic problem. Then of course it comes the response. It can be used, so if you do the pharma testing, we can use this knowledge for inclusion or exclusion criteria when the metabolic pathway of drug is done. But on the other hand, if you are going to do exploratory study, we have no idea about what is the drug metabolism, enzyme, all these things. Here, we cannot use the pharmacogenetic principle, because then it will be biased, we will be missing many things. So in case of exploratory studies, whether it is phase 1 or phase 2, whatever may be the thing, pharmacogenomics has no role. Only after we get some idea about the metabolic pathway, all those things, then we can include the pharmacogenomics. Next slide. Then come to phase 2 and phase 3 study. The phase 2 trial test if the disease responds to new treatment or not. That is a major test. While phase 3 trials test if the new treatment is better than the standard treatment. Phase 2 and phase 3 trials are confirmatory phase. Patients with disease interest are studied to provide further information on dose ranging. Dose ranging and to establish definitely the benefit risk ratio of the new drug. So if we have some idea in the phase 1, then it can be incorporated in the phase 2 and phase 3 clinical trial also. Next slide, please. The advantages of clinical trial using pharmacogenomic knowledge are many, but there are certain restrictions also there. Pharmacogenomic testing during clinical trials has significantly reduced two important concerns: efficacy and safety. Dose in selection and dose modification, exposure and doses higher than otherwise possible only if you apply the pharmacogenomic knowledge. When you use the pharmacogenomic knowledge, the study subject, number of study subjects will be smaller number relatively, and the duration of the study also will be shorter. So, another advantage is it eliminates unsuitable drug candidate and therapeutic targets at the early stage of the development only, so that the failure at the phase 2 or phase 3 can be avoided. Then optimize risk-benefit profiles, experience the outliers and interpatient variability in pharmacotherapy. Interpatient variability can be reduced, then the reduction in the number of dropouts also can be reduced. Then another benefit is when you use the pharmacogenomic knowledge in our study clinical trials, then we can compare it with the other ethnic populations. Next slide, please. There are few disadvantages, but that's also important. When we apply pharmacogenomic knowledge, we use the selected individuals. So sometimes we may not get inadequate information about potential variability in the drug population. It's not possible to investigate safety and efficacy even lower dose of genotype together. Then multiple enzymes are frequently ignored drug metabolism and transport. But in pharmacogenomics, if you just, it is difficult to test for all the alleles. So that is another limitation. When you use pharmacogenomics, then we become overconfident, we may escalate the doses, and the efficacy also may be higher side we will conclude. Then further, erosion of short-term and long-term safety data. So decision may overlook the importance of pharmacodynamic polymers. So pharmacodynamics is very, very important. And of haplotypes. Next slide, please. Then in this, there is about the specifically for phase 2 and phase 3 trials. And about phase 4 clinical trial, here we analyze the report of adverse reaction with pharmacogenetic test. Most important concept that integrating pharmacogenetic drug development is the fact that rare or delayed ADR are unlikely to be observed during clinical trials. Example, Perexlin-induced neuropathy was not evident during clinical trials. But when you follow them for about 15 or 20 months, the neuropathy side effects become prominent. So it is essential that the pharmacogenetic testing should be done in the phase 4 trial also beyond that benefit. In order to harness the potential benefits, pharmacogenetic studies will have to continue well beyond the approval of a drug into its post-marketing surveillance period. Next slide. Now comes the regulatory issues. Regulatory issues, as I went through the website of CDSO, it is not very clearly, they did not spelled out. So I have to go to the FDA website to see what they are saying. This updated one, yesterday only I went through the website. It requires submission of pharmacogenetic data during drug development. But it is not intended to provide definite evidence of safety or effectiveness. So it is only a recommendation one. So phase 1 and phase 2 studies considered these guidelines are often described as exploratory in that they are not intended to provide definite evidence of safety and the effectiveness needed to support drug approval. Nevertheless, PGX studies can provide mechanistic support and be applied in the design of analysis for later trials, potentially improving their efficiency or likelihood of success if the genetic factor can help predict the likelihood and magnitude of response. Next slide, please. This I already mentioned. Next slide.
Thank you.
This is from FDA recommendation. They come out with a table where pharmacogenetic association have been listed. Section 1, where pharmacogenetic association for the data support therapeutic recommendation for about 60 drugs are given. Then associated with the data indicative potential impact on safety or response, about 20 drugs are listed. Then pharmacogenetic association for the data demonstrating potential impact on pharmacokinetic properties, about 40 drugs are given. This table is being continuously updated. So if you get interested, you can go to the FDA website and see it. Next slide. Then about the drug labeling. Drug labeling, when I was a student, this time it was few only. Now the table shows pharmacogenomic biomarkers in drug labeling has to be there at least 570 drugs are listed there. It has a long file. File is 3.67 MB. And the list is they go on adding this. Next one. Then the FDA, that is FDA, European, and the PMDA, that is Japan, all expect the sponsors fully investigate the potential effect of polymorphism on any enzymes or transport that play a major role in drug metabolism or clearance. All three agencies also recommend that examining the effect of genetic polymorphism on pharmacodynamic endpoints. Mostly we read about the pharmacokinetics, but pharmacodynamics should be considered. In general, it is recommended that if genotypes of predictive or drug effects in early trials, enrichment or stratification strategies should be considered to increase the representation of genotype of interest in subsequent patent studies. Although the guidelines on pharmacogenomic, pharmacokinetic research have matured rapidly over the last few years, those surrounding pharmacogenomic, pharmacodynamic assessment are considerably less well developed. Next slide, please. This is the CDS are such. Finally, only this finish I could get. Whenever they give permission to any drug company to carry out the clinical trial, this is the advice they give. At least 50% sites from government sites. But second part is important. The firm should comply with the requirements for sampling of DNA biomarkers for pharmacogenomic study and shipping to other countries. This is the only advice they give to the sponsors. All the other things are about this committee, other things. So the relatively CDSO is not giving so much important for pharmacogenomics in drug development. Maybe I made a mistake in the yesterday website. This is what I could get together. Next slide. Generally, DNA sample collection also there are certain guidelines for clinical trial. From an operational perspective, DNA samples must be collected before genomic work starts. A recent survey showed, this is from Western data, 80% of the companies routinely collect DNA samples from subjects for phase 1, phase 2, and phase 3 studies. Approximately 20% companies routinely collect DNA samples as a part of phase 4 studies. However, support for pharmacogenomic sample collection is not universal. For example, Brazil and Thailand, they say don't collect any DNA sample during clinical trial. China says yes, you can collect but do not transport to outside China. So these are the variabilities there. I think I understand from the previous slide that India is not permitting the transport of DNA outside India, but they will have to follow certain guidelines. Next slide. So to conclude, pharmacogenomics holds the promise to drive most efficient, effective drug development. Incorporating pharmacogenomics into drug development is associated with a number of challenges. Some are being addressed, whereas others still need to be addressed. It is often difficult to recruit enough patients when studying small genetically defined subgroup for patients. Hence, novel trial designs that allow smaller sample size and faster enrollment is needed. A number of alternative trial designs, like adaptive clinical trials, randomization, delayed start, early escape, or such a alternative trial designs may overcome the limitations of sample size requirement for pharmacogenomic study incorporation in the clinical trial. Each trial design comes with a specific advantage and limitation. So careful selection of the appropriate study design is very, very crucial if you are planning to use pharmacogenomics in your clinical trial. Then most important is we need collaboration between industry, academia, and the regulatory agencies to improve the success of applying pharmacogenomics in drug development. These are the things I want to share. I thank the opportunity. Again, next slide.
Thank you, sir.
I thank you very much for giving opportunity to share my experience. Actually, almost for 5 years I did not attend any conference. This is a chance to look at the younger faces. Thanks a lot. Thank you.
Thank you, sir, for your nice deliberation. So this is open for discussion. One or two questions from the audience.
Yes, please.
Any question? No question. Thank you all. Thank you, sir.
Okay, thank you very much. I again thank the organizers, especially Biswas, Tripadi, and other organized secretaries for giving the opportunity. Thanks a lot.
Thank you, sir.
Bye.
Thank you very much. We had an exciting opener to this session. Now we go on to another very important aspect in clinical drug development, which is setting up a BAB study center, challenges and solutions. It will be tackled by none other than Dr. Usharani Pingali, who heads the Department of Clinical Pharmacology and Therapeutics at the Nizam's Institute of Medical Sciences in Hyderabad. So over to you, madam.
Yeah. I thank the organizers, especially Dr. Biswas, Dr. Tripathi, and of course Shambhu for giving me this opportunity. I hope my slides are visible and I'm audible.
Yes, audible.
Yeah, okay. So the topic given to me was setting up a BAB center with challenges and solutions. And and this has been pretty close to our heart because we've done the BAB studies for for quite a long time. So I would just like to give my presentation with a little introduction. The current BAB scenario in India, the regulatory requirements for setting up a BAB center, what are the potential challenges, the key solutions, and the small take-home message. Of course, we know that bioavailability refers to the rate and extent to which an active moiety or a drug or metabolite enters the systemic circulation, thereby ascertaining its action. And that is defined by actually 3 pharmacokinetic parameters, AUC, Cmax, Tmax, whereas bioequivalence is actually done to establish the absence of statistically significant difference in the rate and extent of absorption between the reference and the test product as per the new NDCT rules. So for this, we need to have some bioavailability and bioequivalence centers to do BAB studies, and the centralized registration and submission is done using the SUGAM portal, and of course we have good regulatory guidelines have been laid down, and we need to follow some minor country-specific variations when we are doing it for other countries also. So we have actually 3 pillars for doing BAB studies. The main one being the clinical pharmacology unit, then we have the bioanalytical unit and the quality assurance unit, and all the 3 need to function hand in hand, otherwise studies will go haywire. So we have some regulatory requirements for setting up BAB centers, which very clearly states that they should be registered with the CDSO by applying through a CDSO form, and you need to have some payment being made, and they should have an in-house or associated ethics committee. It can be an independent ethics committee or an institutional ethics committee. However, the more important aspect is the oversight on the bioavailability and the BAB studies at a center by the ethics committee, which is giving approval. So this is one thing which we have seen has been lacking in some of the BAB centers, where the ethics committee members don't oversee what is actually happening. And then these city rules actually go ahead saying that we have detailed description on how to apply for a BAB center. We have various chapters which discuss the requirements, the registration and the application, how to import the drugs, and how to actually do the analysis, the applications for that, and the guidelines for conducting. However, we are more concerned with the registration, because all the BAB centers need to be registered, for which you have to apply using the CT form. So which, if everything is fine, you get an approval with the CT09 form, which is valid for 5 years. But if there are some objections, then it is, you need to actually fulfill the corrective procedures you need to do and then resubmit. Or it may be even rejected within 60 days of application, where you can again apply to the central government, and then you can appeal to the central committee and then give your response within 60 days of an appeal. So when you set up a clinical BAB center, the clinical pharmacology department, the clinical pharmacology unit is something which is very important. That is the basic point for doing such studies, and every time you need to have an access control to the entrance, which is once the bag and baggage check is done and the volunteer is sent inside, then they should not come out again unless there is a specific requirement, because everything is there within the unit. Like you need to have washrooms, and one thing which you need to remember is the washrooms should not have a latch from inside. It is only that you can just close the door, but you should not have latches inside, because if there is an emergency, then the volunteer needs to be accessed. So, and then you should have some sort of a calling bell, which we used to have in our unit, where if there's an emergency, the volunteer can press the calling bell and someone from outside can come in. There should be a good dining area for the subjects, because they will be housed for nearly 24 to 48 hours or even more, depending on the drug. The dosing area needs to be clearly defined with the proper seating, spacing and all that. And then you should have an area for drug supplies, and that should be under the control of the pharmacist. And sometimes we have seen in some places, the drug supply area is kept within the unit where the dosing is being done, and perhaps that is not a very good procedure. Then you have a medical examination room to see the volunteer at time of screening and all that. And then you should have some area, a small area for certification, sample separation, where the samples are separate, centrifuge separated, and then they are stored in a deep freezer for before analysis is being done. And then it's always advisable to have an entertainment area for the volunteers, because once they're housed, then they need to have some sort of an entertainment. But that should not include any heavy physical activity, so it should be something simple. And one important thing is, between the beds, again, there should be adequate space. I think ideally says between 1 to 2 meters or 4 to 5 feet, because if there is an emergency, there should be space for movement of the crash trolley or any emergency equipment between the beds. So if you have them very close to each other, then that's going to hamper the emergency process. So you should have adequate spacing, good nursing staff, qualified physicians to look after the volunteers. Then you should have, first I think this is what I've covered, the pharmacy lab, then the volunteer housing area, the volunteer verification of the participation. So we used to give them ID cards. So every volunteer should have some sort of an identification, because many a times the volunteers replace each other, so that's not very advisable. And then a well-equipped ICU or emergency procedure area. Then when you go to the bioanalytical requirement, again the bioanalytical lab, which can be within the unit or it may be outsourced space. But the bioanalytical unit ideally should be standardized and regulated, and then they should be all validated equipment with strict SOPs which are in place. SOPs need to be there for clinical investigation aspect, for volunteer housing, and for everything right from the screening procedures till the volunteer discharge. Similarly, even the bioanalytical unit needs to have proper SOPs, again which were lacking when we had gone for a couple of inspections. There were SOPs, but the people were not trained on the SOPs, so that's another issue. So there should be a training certificate for all the staff on the clinical side as well as the bioanalytical side. So the bioanalytical equipment, here we have the SOPs for method development, validated standardization, and so on. So if we don't follow the regulatory requirements, then we are always we've been in the news for the wrong things. So that is the flip side of the whole story. So we should need to take precautions that we don't come into the news for the wrong things. And to oversee all this, we need to have a good quality assurance of the quality control unit. So the QA/QC systems with written SOPs, they need to monitor and oversee the whole procedure being conducted, whether it's being done in accordance with GCP, GLP, GMP, and applicable regulatory requirements. So this is something which is very important. So the QC system needs to take care of all this, especially at the GCP part when the volunteers being housed. And the QA personnel, they should be independent and they should assist in quality control, including conducting and monitoring trials. So why we say it should be independent is if one inspection we've seen that one of the clinical investigators, colleague or friend, they were actually doing the QA aspect. So that is not very advisable because then it's not done the right way. So and then documentation needs to be there for a QA person. So monitoring should be done at that time when the studies are being conducted. Many a times the monitoring is done after the study is being conducted. So then there are chances that you may miss a certain aspect. So during the study it should be done. And then even for the bioanalysis, because this has also been a topic of apprehension and debate. So bioanalysis again is something important. And then performing reporting and pharmacokinetic statistical analysis also should be taken care of. Ensure that all the software systems and instruments are validated and the systems are in place. So this should be again, software accreditation and everything should be preferably done online with audit trail so that everything can be tracked very efficiently. And then the quality management system should also have, we should be able to do the root cause analysis, tracking the trends and seeing the data integrity and whether it is, and if there is any issue, after they do the root cause analysis, they should do the Tapas or the corrective, appropriate corrective and prevention action should be taken. But one thing which we have seen is if the databases are not very well maintained for the volunteers, then there are chances of volunteers being involved in many clinical trials. So some systems should be set up at the individual unit level as to track the volunteers also. Then MOUs with any accredited laboratories should be there ideally because we have seen that some of the times the sample for screening was sent to different labs. So each time we are sending to a different labs, there is again a chance that there is lab-to-lab variation. So what may be normal in one lab may not be normal in another lab. So this was one thing which needs to be taken care of and for which you should need to have an MOU detailing the process of the analysis also for those lab tests when you are actually screening the volunteers. So that was missing in some units. And independent ethics committee. So this was again one thing, the most of the stuff people had an in-house ethics committee saying it's independent ethics committee, but then monitoring was not being done on a regular basis, which again raises the cause for cause inspection by the regulators. So as per the regulations, we know it should be within the 50 kilometers radius of the study site. And of course, biological lab we have discussed, and then there should be provision for emergency services, especially if you are doing with some drugs which you think are going to be problematic, the newer maps and stuff like that. There should be some sort of a system to transport the patient to an emergency place if the local unit is not equipped properly with the emergency handling procedures. And that should have proper SOPs, which again were lacking in some organizations. Documental, archival and retrieval. This is again very important. We have been talking of SOPs all through. And SOPs, just having SOPs is not fair enough. We should be trained and then that should be used as per the SOPs. So documentation, we know if it's not documented, it did not happen as per the GCP guidelines. So everything needs to be documented real time. So this was again lacking when we have visited some units because patient came yesterday at 9 o'clock is not okay. The patient has come at 9 o'clock, it should be documented the same day. So then it's going to be an audit finding. Then the archival room should be again totally access controlled with the delegation of responsibility being given to the person who's going to actually have an access to the archival facility should be a fireproof, waterproof, termite-proof room with proper temperature, humidity control. And now since we are having most of the data in soft copies or which is actually either some provision should be there for the hard disk or the pen drives or wherever the data is being stored. There should be some SOPs, and then that should also have some sort of a system where you can take out the data. And then retention of the product. So it is once the study is done, the documents and the products have to be retained, so there should be SOPs for that. The SOPs are something which is extremely important, and for retention of the product, we've seen that a couple of centers did not have a proper SOP because you need to retain the product which was used with the test and the standard product 5 years after conduct of the study or 1 year after expiry of the drug, whichever is later. So that is little important. So when you see the flow of the events, you have the principal investigator who again has most of the time the protocol is given by the sponsor, but the protocol should be agreed upon by the investigator only. They say that we agree to this protocol, the study should start. Then it's approved by the ethics committee and the regulators. Then only after that the consent is taken, the volunteers are screened. Then the flow continues till the study is completed. The volunteer is discharged and in good health. And then there should be ethics committee oversight throughout the study. The reasons for cause inspection for bioequivalence studies, and especially when you go to the FDA website, they have enumerated very clearly, and most importantly what I've seen was they have talked about data integrity and validity issue, which was there in about 60% of the inspection. So data is something extremely important, and validity of the data. So it should be acceptable in all aspects. Then only the study is going to be a successful study. Of course, the others are there, but the percentage is relatively lower. So most important, 60% is data integrity issues. So yeah, what are the challenges? And I'll try to finish a little faster. The challenges first, let us take the infrastructure and manpower. You have very good SOPs, you have very good systems in place, but you need to have proper manpower and infrastructure. Infrastructure, a very good clinical pharmacology unit, having, as I discussed, the bed spaces between the beds, the environment, and then there are very clear-cut guidelines laid down by the FDA, WHO, and CDSO on how to actually create the unit and what is the requirements for a unit. And then you should build a system such that, the volunteers, the second thing is the volunteer transport, as I already mentioned. If there is a crisis, there should be a system in place where you can immediately help the volunteers doing some immediate CPR procedures and then transfer the subject to the nearest ICU. So ideally everything should be in the same flow so that you don't waste time. And then recruit qualified personnel to conduct BAB studies. So many a times you have seen the people looking after the DSP studies not being doctors. So it's very difficult for them to handle if there is an ADR or an emergency. So ideally the trained personnel should be there, and if you're doing phase 1 or any other BAB studies with some drugs which are really critically known to cause adverse drug reactions, then you should have even an anesthetist. Ideally, like we used to have an anesthetist posted when we had such studies going on. And then organogram, which clearly defines the roles and responsibilities. So many of times this is missing. That's what we have seen. So then the blame game starts as to this is not my role and I wasn't informed. So if you have a clear organogram and a delegation of responsibility log, then most of can be actually taken care of. Equipment, the equipment needs to be actually validated, even including the BP apparatus. We have seen the ECG machine, they are not being calibrated and validated. So unless it is not calibrated properly, and if there is an expiry in the calibration, please redo the calibration, including the height, weight, everything which is being used, as well as analytical equipment needs to be calibrated and validated. And then there should be SOPs for operating, which again needs to be, people should need to be trained on that. Like if a nurse is performing the height and the weight, then you should have an SOP and then she should be trained on taking care of all that. And then weekly and monthly maintenance of the equipment as per the requirement. And then what we have had a couple of instances in our own state where the volunteers participated in multiple clinical BAB studies, and then there were a lot of issues, and was one of the members in that committee. So what suggestion we had given is that it should be an Aadhaar-based, system for registration of the volunteers. So the moment you actually click on that Aadhaar number, then there should be some way like a red light or something glowing which tell you the volunteer participated in the study within 3 months, because we know that at least 3 months should be there between participation of 2 studies. So that was something which we have suggested. So there should be some sort of a system in place where you can track that the volunteers have, not in multiple studies. Training and SOPs, this we've been, I've said a couple of times, there should be training on the SOPs to all the people involved in the studies. And then there should be an audit trail, especially for major operations. You should be able to find out who made the changes, when the changes were made, or how the changes were made, and everything you should be able to track retrospectively what all happened. So that's an audit trail, and that's again going to be very helpful, especially for volunteer studies when there are issues. To have systems in place to handle medical emergencies, which we have already discussed, and then there should be drills conducted, like we used to do some mock exercises before starting the phase 1 clinical trial, because that was, I think we were one of the people to do first the IV formulation phase 1 also we have done. So there should be some sort of a training to given to all the staff. Data security, yes, this is something again very important and again a big challenge, because systems should be such that there is security because access, which means that the data should have an access control. So even if anyone is opening, people should not be able to open the laptops or the place where you have data stored. We should be access controlled, and then there should be passwords. Again, there should be delegation of responsibility for who is going to have the passwords. The soft copy, especially the IT, should be very, very strong, and there should be separate servers which are going to store the data, and then they should be able to retrieve the data. You cannot store the data and then say the system's crashed, which was one center which had given us such an answer that the systems have crashed. There should be backup of the systems again, so that this doesn't happen, and then if there is an inspection or an audit, they are able to track it. So you should build systems for archival and retrieval, hard copies, soft copies, everything in the shortest possible time. This was again another issue, because research is in a place which is around 15 kilometers from the unit. So I think, if you are storing in such a place, then there should be some provision to get the data immediately when there is an audit, because many a times the audits are not informed prior. So. And partnerships and triumphs, as we discussed, conduct audits with the labs before finalizing, and then choose the labs, which is an agreement to the principals of the BAB centers, and then of course take it to the CDSO registrations. So I think we're kind of getting over. So some points to take home. When you set up a BAB center, it is very interesting, very challenging, but of course it is rewarding also when you do BAB studies, because that acts as the training for the students, because once they leave the unit, then they may go and join some BAB centers as undertakers or the head of the unit. So that will be an experience and an exposure to them. And then focus on getting the training and the system in place before applying for CDSO registration. Conduct mock drills to ensure that everyone is trained on the SOPs and adhere to the SOPs. Ensure that all equipments are calibrated and maintained, and then you should have a register where you're going to update it regularly, and keep the old calibrated logs also into one file, because that will again be a point of check when there is an audit. Ensure robust documentation, real-time documentation, and proper storage and archival of the documents and a backup retrieval whenever it is required. So these are the references, and thank you for giving me the opportunity, and I would like to thank my student Dr. Rupali who's helped me in making these slides. So thanks a lot, and over to you, Shambhu.
Thank you. Thank you, madam. We all agree that was a wonderful 360-degree coverage of the area of BAB study setup. And you covered everything from the requirements for the center itself, as well as the bioanalytical part, as well as data security and everything. So once again, thank you very much for sparing your valuable time. We will now go on to the last topic of this session.
I think Dr. Sandhya Selvarajan, we can add.
Okay, she has a question. Yeah. Dr. Sandhya.
Yeah, good. Thank you, madam. That was a very nice talk, saying everything about the BAB study. I have a query. For example, if a parent institute is having a branch which is in another city, more than 50 kilometers, can the parent institute have the ethics committee to monitor something which is happening in the branch hospital of that particular parent institute, which is more than 50 kilometers and in another city? Or should we have the ethics committee in the same hospital?
50 kilometers has been, topic of debate when, following the guidelines. Actually, what happens is 50 kilometers is not the hard and the fast rule. You can't measure that. So what we actually mean is, since the ethics committee members have to go and monitor, so if it's within 50 kilometers radius, that was a random number. It can be 60, it can be 40. So if it is within the 50 kilometers radius, then it's easy for them to go, because the ethics committee members again do have a 9 to 4 job. So apart from the job, if they have to go to a center which is around 100 kilometers, then it's going to be difficult. So that's an arbitrary number of 50. So for monitoring whatever is convenient and comfortable to the ethics committee members, yes, within, if it's around 50, 60 kilometers, then they can travel up and down, then it's fair enough. Otherwise, it's always better to have another, ethics committee that's going to monitor things. So that's it.
Thank you, madam.
Thank you, madam. Just,
Subhankar?
hello? You are audible?
Audible.
Subhankar, I think you're muted. You wanted to ask something?
Sir, bolun aap shona jaচ্ছে.
Nice presentation. Just I want to know from you that in case of your BAB center, so there is so many components. So one of the most important component, that is your analytical laboratory, and it should be approved. So if it is not approved, but the analysis of the drug is being done following the principles of GLP or NABL, so whether the data will be accepted or not.
Shambhu, can you repeat? It wasn't very audible.
Sir, apni oikhane theke.
Hello, madam.
Yeah.
So BAB center, so there is so many components. So one of the most important component, that is your analytical laboratory. So for this analytical laboratory, that should be approved by the GLP or NABL or appropriate authority. So if it is not approved, but the system is there, and if we follow the principles of GLP, so in that condition, the data will be accepted or not?
Normally, ideally, they are asking for a regulatory approval for the labs also. Otherwise, then the data may not be accepted. The labs are not approved by the regulators. And that was because there are chances of the data integrity comes into place. So ideally, you should have an approval by the regulators for an analytical lab.
Because you know very well that it is very difficult to maintain this type of laboratory under government organization or institution. Okay, thank you.
Yeah, Professor Shantanu Tripathi for the final comment.
Hello, sir. This is Shantanu Tripathi. So let me share my experience as Dr. Mondal was asking regarding whether the analytical laboratory needs to be also approved. The answer is clear, yes. We recently had some attempt that the volunteer housing is in the hospital, okay, unit, BAB unit, it has a volunteer housing and sample collection, okay, dosing and then sample collection and then its analysis. So analytical part is definitely difficult, and many a times the hospital that intends to start this center, they find it difficult to invest into a high-end laboratory. So we actually tried in Kolkata that the hospital agrees to do the volunteer housing and sample collection, etc., and transporting the samples to another facility which is maybe 20 kilometers away, and there the analysis will be done. But then we are very clearly told by CDSCO that both these centers needs to be approved by them, and we actually went for the hospital will take a couple of years to develop their own laboratory. But then we got the response that in that case, again you have to face the approval from the CDSCO. They will inspect, and under the same roof, whether we have the volunteer housing as well as the analytical laboratory. So this is one experience. So we are waiting for the same hospital to develop its own laboratory setup where the analysis can be done under the same roof. This is one experience. The other is in this city in Kolkata. There is already one center which is running for a good number of years, and there, because of the logistic reasons, they have the voluntary housing in one place, and maybe in another 500 meters away, half a kilometer away, there is the analytical laboratory. So whenever the CDSCO inspection team comes, assessment team, they visit both these centers and try to see whether that is as per the requirement, the regulatory requirements. So it should be very clear that if you want to do BAB, it has both these two components, and the third component, of course, you haven't discussed about the ethics committee. The ethics committee could be an independent committee, or many a times the center itself has its own ethics committee, BAB center itself. But that center, even if it is within that unit, it also should maintain its autonomy and independence. So these are the three components of BAB center. I think you agree, Dr. Usha, what we just discussed. Over to you. Thank you.
Yes, sir, that's very correct, because now when you're applying the DCT of game approval, they will say the clinical part is being done there, and which lab is going to do the analysis. So in the clinical trial, the same thing they follow. So unless it is approved by them, then they don't give approval for even conducting of the study. So they need to be approved by the regulators, sir.
Thank you very much, Madam. That was wonderful. We now go on to the last talk of this session, a very important topic, ethics in clinical research, neglected issues. And we have a change in speaker. We have Dr. Shreyashi Dasgupta from the School of Tropical Medicine in Kolkata to speak on this.
By the time Dr. Shreyashi gets ready, I think there is a hand somewhere.
Okay, Dr. Sinha.
Sir, you may go ahead with your question. Dr. Sinha.
No, I think partly it is answered regarding the analytical facility. I think what I wanted to know is that there are two types of lab facilities that need to be seen. One is the diagnostic lab, which is meant for the lab biochemistry and all that. That should be also accredited, and if this data needs to be submitted to the, generally for industrial BAB centers, you need even the central, what we call the central lab, to also have some accreditation. The bioanalytical lab actually need not be there in, many pharma companies, when they engage the centers, the academic, the medical center, college centers, they use that mainly for the clinical part. because their bioanalytical methods have been already developed by the company, and they have they have the center of the CDSU with them. That is one part of the story. But the second part of the story, as yesterday also I was talking about the phase 1 centers which have been approved recently by IPMR, which is also similar facility. There, when we developed the phase 1 center, especially to encourage the Indian new drug developers, the research institutes to develop new NCs, there I think the bioanalytical facilities are lacking. And none of these centers which have a facility to begin with. So, bioanalytical facility approvals understood, but it is more for BAB centers it is not really required, because most of the BABs are done for regulatory purposes by the pharma companies, and they already have that approved centers with them. It was a comment more than a question. Question was also on the ethics committee. These phase 1 studies or bioequivalence studies, these are non-therapeutic in nature, primarily clinical pharmacology in nature. So, is there any additional clinical pharmacologist that should be included in the committees governing? They often govern both BAB as well as phase trials, but especially the BABs and phase 1, should they have a clinical pharmacologist on their board as an additional member study, at least for these studies, beyond the approved composition, or maybe add for an additional approval for this?
Would Dr. Usha take the question?
As a guest they can come, because if the clinical pharmacologist is the basic scientist as per if that has been shown, then it is fair enough. But if you have a basic scientist and the clinical pharmacologist is called as expert, they can only visit, they can only give their comments, but they can't participate in the decision making if they are not part of the ethics committee. So you can take their expert opinion, there is no doubt about that, but that should again be in your SOP.
Thank you, Dr. Sinha, and thank you, Professor Usharani Pingali, Madam, for this wonderful deliberation. And now we are requesting Professor Shantanu Munshi to kindly felicitate our chairperson, because sir has another meeting, emergency meeting now. So, Professor Subhankar Chowdhury, sir, kindly felicitate.
Good afternoon again. This is a momentous occasion for me to be sharing the platform with stalwarts like C. Adithan sir and Usharani ma'am. And I believe this topic was to be discussed by Dr. Urmila Thatte madam, who is one of the pioneers in research ethics in the country, and I shall make a humble attempt to cover some of these areas. And before I begin, I would like to acknowledge the inputs from my mentor, Professor S.K. Tripathi sir, and some of my seniors who have helped me in preparing this presentation. So, what I will be speaking today is I'll try to dwell upon the neglected areas when it comes to research ethics in our country. So, to discuss the same, I have thought of the whole thing as I have divided it into 4 perspectives. So, I will be looking at the neglected areas from the perspective of conduct of clinical research, from the perspective of an ethics committee, from the investigator's perspective, and from the site administration's perspective. So, when we talk of research, or when we talk of ethics per se, it is basically the differentiation between what is right and what is wrong. But when we talk about right and wrong, there is a gray space in between where we actually are not sure which is the right way to go forward to. So, coming to conduct of clinical research, if we look at the National Ethical Guidelines for Biomedical and Health Research involving human participants, the ICMR guideline, which was released in 2017, so there we find that when it talks about academic trials, especially for compensation related to research harm arising out of academic clinical trials, it says that research participants who suffer direct physical, psychological, social, legal or economic harm as a result of their participation are entitled, after due assessment, to financial or other assistance to compensate them equitably for any temporary or permanent impairment or disability. So in this case, participants, in case of death, participants' dependents are entitled to financial compensation, and it only says that the research proposal should have an inbuilt provision for mitigating research-related harm. Now, when we look at compensation, in the new drugs and clinical trial rules, when we talk about clinical trials with new, for investigational new drugs, or even for BA/BE studies, there is a clear rule or clear guideline as to how to go about compensating the participants who suffer harm out of these research. How that compensation is to be calculated, what is the pathway, or what is the way in which such compensation is to be given to them, how the analysis, causality analysis is to be done. But when we come to academic trials, there is no clear guideline as to what the quantum of compensation should be. Whether it should be aligned to that which a clinical trial for, participant in a clinical trial for a new drug is entitled to get, or whether because this is an academic clinical trial with a drug which is already established, which is already being used by other people in the market, can we say that they maybe are entitled to a lower quantum of compensation? There is no clear consensus on this, and this is one area in which we need further clarification or further guidelines or further inputs from the regulators. Now coming to standard of care. Now when we talk of standard of care, if we look at the global picture of it, there is a wide disparity in when we talk about access to standard of care. Well, what is a standard of care in maybe a developed nation like the United States of America may not be accessible to the average population over here in India. So when we talk about a multinational trial or a global clinical trial, can we have a similar standard of care for all participants, whether they belong to a lower income or middle income country such as ours, or when we talk about the developed countries? And supposing even if the sponsor is willing to offer the standard of care as part of their clinical trial, what happens to the participant after the trial is over? Now, while the participant was a part of the clinical trial, he or she was getting the standard of care as due to him. But at the end of the trial, when the trial ends, the participant is no longer able to afford that standard of care. He was getting it because he was a part of the trial. What happens to that participant after it? Have we gone through that? Have we looked into the ethics of that part? Now, and in some cases, we do not have a clear consensus on what the gold standard is. What is the gold standard for standard of care, so as to say? Especially in oncology, there is no consensus. There is in some cases a lack of consensus among the physicians on what the standard of care should be. As was being talked about by Dr. Shovaldash yesterday, even in lifestyle intervention trials, we do not have a gold standard. We do not know what is the advocated standard of care for a particular lifestyle intervention. So, this is also an area which needs further deliberation. A similar or related topic is a post-trial access. Now, for the sake of students who are here, post-trial access is when you have a new drug or an investigational new drug which is being given to a trial subject as part of the trial. Now, after the trial is completed, the investigator feels that the participant would benefit out of this new investigation, out of this investigational new drug, and he or she should be provided this drug because he has shown a clear benefit out of it. Now, this can only be given if there is no other standard of care or no alternative treatment available for that particular condition. And there are other rules and regulations how we need to procure this post-trial access. The sponsor is actually responsible for providing the post-trial access, but is not liable for any adverse effects arising out of it. Now, who takes care of these adverse effects, even if they arise? Because the sponsor is clearly not liable. Is the investigator liable because he has vouched for the participant in this case? Also, for how long can this post-trial access be provided? For how long would a sponsor be willing to provide such a post-trial access, especially if we are talking about biologics? Now, in that case, because these are very costly drugs, so to say, so how long can this, can we almost force a sponsor to maybe provide the post-trial access? Do we have any clear-cut guidelines on that? And again, as I was saying, what happens to the trial subject after the trial ends? Suppose we have maybe for a simple condition, say for diabetes, we have two drugs. We are doing a superiority trial and we have drug A and drug B. Drug A is the standard of care, drug B is the new drug, the investigational new drug, and at the end of the trial we find that patients who were receiving drug B have improved significantly, or their glycemic parameters have improved significantly by use of this investigational drug. Now, at the end of the trial, can we ask the subjects to go back to maybe drug A, to which they were not responding? Or because we cannot expect the drug to come into the market immediately. There will be significant amount of time after which the drug will be available in the market. What about their medical care during that intervening period? And if at all the drug comes to the market, will the patient be able to afford the drug? Because most of the patients who participate in clinical trials in our country are from the lower socioeconomic class. So what happens to them later on? Next, coming to the informed consent process. Now, in spite of all the good clinical practice workshops, trainings, etc., can we truly say that the informed consent process in our country is an informed one? Do the subjects actually have the wherewithal to fully understand what they are getting into? Can we improvise this method further? Next is the topic of, or the term of deferred consent. Now, this was a term which came to the fore during the pandemic, because in many cases we have patients who are not able to give consent at that point of time, but we have investigational drugs which the investigator feels that the patient may benefit out of. Even in case of terminally ill patients or end-of-life care also, the consent has to be deferred. We need to put that patient into maybe an investigational arm where we feel that the subject would benefit, but only after the subject regains consciousness can we take the consent from the subject. So we do not have any clear-cut guidelines on how to go about taking a deferred consent, who are the people or in which conditions we can take it, how we should take it, how we should document it. All of these are not available in the guidelines at present. There is a need to incorporate them because we never know when the next pandemic will strike us or when some other emergency comes up. Also in terminally ill or end-of-life care, in some cases the patient is not able to decide for himself or herself. It is the patient's family members who decide. So does the patient at all wish to continue with a drug which will save his or her life, or does the patient not want to take or not want to bear any more of the condition that he is in? We do not know, and this is probably another area where we can have more discussion on. Some other concerns, while we are talking of conduct of research, is especially in surgery, we have sham surgeries or sham procedures to account for the, to have a similarity in both the groups. So in one group we are doing a surgery, other group we are doing a sham surgery, so that there is adequate blinding, or the people, we have comparability between the two groups. Now, can we truly assign or subject somebody to a surgery which that person does not require? I do not know how much this is being done nowadays, but this is also a thing where we should delve into. Now, there are some drug trials which have a lead-in group. I had come across a study protocol a few days back where they were trying to treat hepatitis C patients. Now, hepatitis C has a treatment as of now. Now, this was a phase 2 study where they wished to start a group on an investigational new drug, naive hepatitis C patients, and after a certain period of time, if they do not find virologic, if the patient does not respond adequately, they will not continue with the study anymore. They will terminate the study at that point of time. Now, these patients have enrolled themselves into the trial with an expectation that they will be cured of their condition. Or when in the first place when they came to the investigator, at that point of time also they had an inherent wish to be cured of their condition. Now we have incorporated them into the trial, asked them to take a drug. At the end of the trial, we are saying that no, we have not been able to cure you. Now you are left to yourself. Now you decide what treatment you are about to get. Now, is it not the responsibility of the investigator or the sponsor to ensure that such a subject gets the treatment which is already available? So, we do not actually, these things are not discussed in the guidelines as of now. So I feel we need to incorporate these two. Now, again, washout periods in trials. We have crossover trials where we have subjects in arm A and arm B, and after they continue with the drug till some point of time, then we have a washout period so that the effect of that drug is washed off, and then we cross them over to the opposite arms. Now, during this period of time, during this washout period of time, they are not on any therapy as such. So, is that actually ethical? Next is decentralized clinical trials. This is coming up in a big way nowadays, and I feel as a country we should also incorporate adequate guidelines as to how to go about conducting decentralized clinical trials, because if we have to compete with other countries, this should be incorporated in our regulations also. Next, just a few points about the ethics committee. So we have several ethics committee over here, but this is something which is observed that in the review process of the ethics committee, the non-medical members usually are reluctant to participate, or they do not find themselves able to express their feelings or express their view as openly as the medical members are able to. So we need to give further impetus to the non-medical members so that their contribution to the ethics committee is also duly recognized. Reluctance to engage in continuing review process. So we have ethics committees, we have GCP certificates, but do we have continuing review process? Are we taking this as seriously as it should be? Or are we continually reviewing the proposals that are submitted to us? After approval, do we go about actually looking into how those studies have progressed, whether they have actually published their results, how they have collected the data? So this overseeing, the ethics committee is supposed to oversee the entire research conduct. It is not just only about approving studies, but after approving, whatever role is should be conducted by the ethics committee, are they doing it actually? And do we have any other means to actually see whether these ethics committees are doing what they should be doing? Are they addressing the conflict of interest adequately, and is it being managed as it should be? And absence of continuous capacity building activities. So SOPs are updated, but and maybe these are even circulated to all the members, but adequate training sessions for SOPs, or maybe recognizing that newer guidelines or newer updates to the guidelines are coming up, and appraising the members about these developments. This is not being done on a regular basis, and I believe this should be looked into. Now, when we talk of investigators, we need a GCP certificate before we do any kind of a clinical research, but that gets confined to only having a documentation of a GCP training. So we are, most of the investigators have a general tendency to just mechanically comply with the minimum ethical standards that are required of them. They do not actually delve into the intricacies of these guidelines or the ethics related to research. So there should be some mechanism by which this can be further reinforced. And when we come to site administration, many a times we find that there is a lack of insight among the site administrators in terms of ethical conduct of clinical trials, ethics related regulations, guidelines, etc. Also, they are in most cases oblivious to, or they have a tendency to ignore their application to the trial subjects in respect to medical management of subjects when they report with any adverse reactions, etc. So this is just my penultimate slide. So I just wish to say that in lines of the famous ethicist Michael Josephson, so he had said that ethics is not about the way things are, it is about the way things ought to be. So taking a cue from this, I would just like to say that we should not be comfortable in the gray areas, but these are actually the areas in which we should work upon so that we can further refine the guidelines that are present in our country at the moment, so that we can actually conduct good research.
Thank you, Dr. Shreyashi, for your nice presentation. Now the topic is open for discussion. One or two questions from the audience. So if there is no questions, so thank you, Dr. Shreyashi.
Thank you so much, sir.
So now we are going to conclude this symposium, clinical research and ethics. We had 3 speakers at this symposium. Dr. C. Adithan, who has nicely described the pharmacogenomics to support drug development research. Dr. Usha, she also discussed on BAB study center, their guidelines, etc., etc. And lastly, Dr. Shreyashi, she informed regarding the ethics in clinical research, and these 3 speakers nicely delivered their topic. So I will I thanks to all of you on behalf of the chairs, myself and Professor Subhankar Chowdhury. Thanks to the audience as well as the organizer to cooperate to organize this symposium. Thank you.
Thank you, sir. Thank you, Professor Usharani Pingali, madam. We miss your physical presence, but thank you for your lecture. So now may I request Dr. Shoibal Mukherjee, sir, to kindly felicitate our respected chair, Professor Tapan Mondal, sir.
Also may I request Dr.
Shreyashi Dasgupta to please come.
Once more. Set ready,